All right, given that we got cut off a little bit there at the end of class on Wednesday, I just wanted to take an opportunity to screencast the last example I was going to talk about in class. This way we can keep more on track with our lectures, and I can pick up with some new material on Friday. And so I'm picking up with the research example that I was talking about from some work of my own that I did in northeastern Tanzania. And the question that we were interested in was whether or not the effects of predators early in ontogeny, early in development, shape encounters with larger predators. Now, in particular, we're interested in whether the density effects, consumption by early predators, or the effects of early predators on the traits of organisms as they move into later life history stages changes encounters with later predators. In particular, we're interested in whether the effects of these early predators result in prey surviving better in subsequent life history stages. And so fewer would be killed in later life stages than we would expect in the absence of the egg predator. And so this would be a risk reduction scenario. Alternatively, we might find that the effects of egg predators early in life make the prey more vulnerable in later life history stages. And this would be what we would refer to as a risk enhancement scenario. And this was work that we did looking at a focal prey species, Hyperoleus spinigularis, which is a little tree frog which is found in the mountains of northeastern Tanzania, and it's vulnerable to predators in different life history stages. And we already talked a bit in class about how I went about characterizing the density effect on the eggs of this species. We did transects, where we monitored the production of eggs at the pond through the breeding season, and then we followed the fates of these clutches, as you can see here, catching the tadpoles as they dropped into the cups where they would have been dropping into the pond. And using this approach, we were able to estimate the number of eggs that were laid over the breeding season and their fates. And using this approach, we estimated that as many as 60% of the eggs are attacked by this particular predator, another tree frog, Aphrixlus fornacini, and that this tree frog consumes about 70% of the eggs within the clutches that it attacks. And with these data, we were able to play the thought experiment of determining how many eggs would drop, how many tadpoles would drop from the trees down into the pond if we were to essentially reach in and pluck this tree frog predator out of the system. So we currently have tadpole rain, number of tadpoles per meter squared per day over the breeding season, that's estimated at this. These are essentially the tadpoles we're catching in our cups. But if we instead assume that the eggs that clutches that were attacked had high survival, then we could estimate that the rain of tadpoles in the absence of the predator could be as much as 60% higher. And this is our estimate of the density effect of this tree frog on tadpoles raining down into the pond. We also talked in class and watched a couple of videos about how egg stage predators attacking clutches of arboreal eggs can cause them to hatch early, and that this has consequences for their encounters, maybe, with predators in later life stages. And what we see in this Hyperoleus spinigularis system is that when eggs are attacked by predators, they hatch as much as 2.5 days earlier, and the tadpoles are about 30% smaller when they hit the water. And so what we're interested in here is we know that we have this important egg predator. It's reducing densities dramatically, and it's changing the size of tadpoles that enter the pond dramatically. And we're interested in, do these density and size effects of egg stage predators change encounters with larval predators? In this case, we're looking at the most common dragonfly in the system, a libellula dragonfly. And how do these different pathways contribute to the number of metamorphs that are produced in the system? What is their relative importance? Do they lead to indirect effects across life history stages? Going into it, we had done some previous experimental work where we looked at how density affects things like larval duration, the time and size of metamorphosis, survivorship, and predation rates. And most of these effects go in the same direction. In general, we expect that reducing the number of tadpoles entering the pond is going to reduce competition. This is going to increase their growth and increase their development rates. And this is going to lead to the tadpoles being larger and spending less time in the water, exposed to aquatic predators. And so in general, we were expecting the density effect of egg stage predators to facilitate tadpoles surviving through the larval stage. And so this is essentially saying that we think the density effect is going to cause risk reduction. It's going to change encounters with larval predators such that they survive better than they would have in the absence of the egg predator effects. We also had some a priori expectations based on experiments about what the effect of hatching early, being smaller, might be. So we look at survival as a function of tadpole size, and we see that it's hump-shaped, where they become vulnerable at intermediate size classes, and then they reach a size refuge out here. And so we think that hatching earlier means that you start in smaller, means that you're out here, you're going to spend more time growing through these vulnerable size classes. And so we think that hatching smaller is going to make you more vulnerable and thus result in risk enhancement. Again, an indirect effect where egg stage predators are facilitating or increasing the vulnerability to larval stage predators. So we have these two egg predator effects, density that we think is going to facilitate survival, and then the size effect, which we think is going to reduce survival. And so they're both happening simultaneously and are acting in opposite directions. So we need an experiment which will enable us to disentangle their relative contributions. And so to try to accomplish this, we did a multifactorial experiment where we have three factors here. We're manipulating the egg predator density effect, the egg predator size effect, and the presence or absence of aquatic predators, a dragonfly. Now, we mimic the density effect by essentially starting these experiments at different initial densities. So we start them at high densities, and so this would be the absence of the egg predator effect. High densities because there was no or very little egg mortality caused by predation, so lots of tadpoles are raining down into the pond. But if we have the egg predator density effect, then we're going to start out at a lower density because lots of eggs were eaten, so not so many tadpoles are raining down into the pond. The size effect we accomplished by stimulating egg masses to hatch early. And so we did this in some cases that we could produce early hatched tadpoles, sort of ourselves. And this enabled us to produce small tadpoles similar to the size of early hatched tadpoles. So that's the presence of the size effect. Similarly, if we let them hatch normally and larger at a later date, we would see that that's the absence of the egg predator effect. And so with these treatments, we basically have all of the treatments that are necessary, that are needed in order to test whether we can predict the combined effects of both predators from the individual effects of these predators. And we also need to know how many tadpoles die in the absence of predators, so that gives us a background estimation. So we have that here. For example, let's take a look at the larval predator treatment. So we have here the presence of the larval predator, yes, but we have no density effect and no size effect. If we look at the egg predator here, treatment 8, we see we have the density effect, the size effect, but no larval predator. So you can see how these 4 treatments reflect the density and combined density and size effects of the predators. Now, one of the challenges, though, is that we have these effects that may be acting in opposite directions, and so that might be masked in this treatment. We don't have the treatments here that would tell us what the individual contributions of the density effect or the individual contributions of the size effect might be. And so we need these additional treatments in order to isolate these particular mechanisms to this overall survival. And so we accomplished this experiment in the field adjacent to the pond where the field measurements were made. We used mesocosms, and so that's essentially small experimental ponds where we set up kind of the backdrop of, in a standardized way, of a pond, and then we mimic or manipulate the factors that we're interested in manipulating. And what we were interested in looking at as our primary response variable was larval survival. So how does larval stage survival in the presence and absence of dragonflies change given the density and density and trait effects of egg stage predators? So here we have the effect of dragonflies given no earlier effects of egg predators, no reduction in density, and no reduction in size. Here's the effect of dragonflies given just the reduced density effect of egg predators, but no change in size. Here's the effect of dragonflies given just the reduced size effect of egg predators, but no reduction in density due to egg predators. Here's the effect of dragonflies given both the reduced density and reduced size, the overall effect of egg predators. And here for reference, we have how well did they do in the absence of either egg or larval predators. In the absence of egg or larval predator effects, we see that they did quite well, about 90% survival, and we see that dragonflies by themselves are quite effective predators. They reduce survivorship from about 90% to about 30%. And what we're interested in with these remaining treatments is whether we see that survivorship is higher than this dotted line or lower than this dotted line, which represents the effect of dragonflies given no earlier effects of egg predators. And so if we see that survival is up here, they're surviving better given the earlier effects of egg predators. So that would be a risk reduction scenario. If they are down in here, that means they're surviving less well given these earlier effects of egg predators. And so that would be a risk reduction type scenario. So first, let's look at the overall effect. What we see is that the combined effects of egg predators on density and size at hatching resulted in a very strong overall risk reduction, to the point where, given the earlier effects of egg predators, tadpoles survived in the presence of dragonfly predators almost as well as if there were no dragonflies there at all, which is a pretty dramatic effect. So given this result, we might think, well, a lot of this is being driven by the expected positive benefits of reduced densities. And indeed, we did find that the reduced densities, lower tadpole rain into the pond, because of egg stage predators, did lead to better survival and risk reduction as we anticipated. Now, we might expect that then there was essentially no effect, a very negligible effect of size, reducing survival. But in fact, we found even the opposite of what we expected, that hatching smaller and less developed also meant that you survived better in the presence of predators. And so this is quite counter to our initial expectations and needs a little bit of further unpacking. So to recap, what we found is that there's intriguing indirect effects acting across life history stages. Egg stage predators are changing encounters with larval stage predators that they don't interact with, that occur in different habitats at different times. Furthermore, when we unpack that a little bit more, we see that both density and size trait type mechanisms are playing an important role. Now, the density mechanism, the consumptive effect of the egg stage predator, reducing the number of tadpoles entering into the pond, acted in the direction that we expected. It led to better survival in the presence of egg predators. Now, intriguingly, we had this counterintuitive result where hatching early and less developed and smaller, which we thought would make you even more tasty to larval stage predators, in fact, also resulted in you being less vulnerable to predators. So let's explore this surprising result a little bit more, given the data we have. So one question that usually I get asked about this work is, well, if they didn't have a cost, there wasn't sort of, hatching early out of the frying pan into the fire, they hatch early, they're more vulnerable. And so that's what essentially maintains the trade-off of hatching early. Because otherwise, why wouldn't you hatch early all the time? If you do better when you hatch early, natural selection should favor you always hatching early. So perhaps the cost occurs somewhere else. So if we look actually at another characteristic of our survivors, looking just at the no predator treatments. So this has nothing to do with sort of reduced competition because predators are thinning down the prey. We see that late hatch tadpoles are, generally speaking, quite a bit larger than early hatch tadpoles. And being larger at metamorphosis is generally thought to be a good thing in amphibians. It's been correlated with earlier age of sexual reproduction, so you're starting to breed earlier. It's often correlated with size at reproduction, and so therefore larger individuals have larger clutch sizes, larger males have better chance of attracting a mate. Larger individuals are less vulnerable to desiccation. There are a lot of possible fitness benefits to being larger at metamorphosis. In this system, there may be additionally a fairly immediate benefit to being a larger size at metamorphosis, because recall that as these froglets are emerging out of the pond onto the floating mat of vegetation in the pond, they often encounter these fishing spiders. And what we see is that small metamorphs, as just typically are associated with the early hatch tadpoles, have lower survival when they encounter spiders than larger tadpoles. Larger metamorphs, rather, are better able to escape spider predators than smaller metamorphs. So this could be a fairly immediate benefit to being a larger metamorphosis at metamorphosis, and it may be a cost associated with hatching early. So if they're smaller and when they emerge, how do we then explain this surprising performance early in life history? Well, We don't have a lot of data to speak to this, but when we look at this sort of question of why do early hatch tadpoles survive better, we look at tadpole size here on the y-axis through time. So essentially we're looking at their growth rates. And what we see is that this reflects the window when they're most vulnerable, that hump in the figure that I showed earlier. And then up here, they're too big to really get attacked by this predator. And what we see is that generally speaking, the small early hatch tadpoles are entering the pond, the vulnerable sizes. They're entering the vulnerable sizes later, and they're leaving them a bit earlier. And they have a steeper slope here on this line, which is essentially an indicator of their growth rate. So it looks like early hatch tadpoles may be exhibiting some sort of a accelerated compensatory growth mechanism. They're growing more rapidly through these early size classes, and therefore they're less vulnerable to the predators that are attacking them in these size classes. Now, this appears to be lost later because, again, as a reminder, they're smaller at metamorphosis. So they grow fast initially, but then that seems to level out, and maybe the big late hatch tadpoles, sort of slow and steady, actually catch up with them and then exceed their size when it comes to time at metamorphosis. But that might explain this sort of surprising result that the early hatch tadpoles did better because maybe they're growing more rapidly through the vulnerable sizes. So just to wrap it up, we've looked at the effects of egg predators on the interactions with larval predators, and we see that there are dramatic indirect effects across life history stages. The effects of egg predators dramatically reduce the vulnerability of tadpoles in the larval stage. And this occurs both through the density mechanism and the size mechanism. Both of those seem to be acting in the same direction to facilitate survival, even though we initially thought that being smaller when you entered into the pond would be a disadvantage, would make you more vulnerable. But in fact, we had the surprising result that they seem to do better, perhaps because hatching early induces this sort of more rapid early growth phenomenon, this compensatory growth, which allows them to accelerate and grow rapidly through these initial vulnerable sizes, but seems to dissipate later in larval stage, such that the late hatch tadpoles catch up with them and emerge as larger metamorphs. So it appears that then the cost associated with that may be smaller size at metamorphosis, the suite of negative fitness consequences to being small at metamorphosis. So you hatch early, you're smaller at metamorphosis, so you have these consequences, including maybe being vulnerable to predators as you emerge out of this pond. But as what this is saying is that essentially the effects of egg stage predators that occur several months earlier in up here in the arboreal environment are transmitted through the prey all the way through one life history stage into the second life history stage. So you have indirect effects across multiple life history stages. So that's a really intriguing example from my point of view about how indirect effects can propagate through different habitats and through different life history stages for organisms that have complex life histories. And again, given the focus of this lecture, that both the density and the trait-mediated effects played an important role in this, but in some cases surprising roles that needed fairly careful experiments in order to tease apart what their effects were. All right, well, we'll wrap it for this session.